when we do a lab in Science 10, and every time we do a lab, you will get a lab handout. On that handout will be, okay, essentially this stuff. I will give you the problem that we are investigating. Okay, there will be a list of materials. There'll be a thing that says identify your variables, generate your hypothesis, record your data in a chart and put it here. That's what we're going to talk about, is how do I assemble this proper lab report so that I get the best mark possible. Okay, now remember, anytime you do a lab report in my class, you can bring it to me ahead of time, okay, before it's due, and I will look it over and give you tips and pointers. Okay, take advantage of that. Okay, you have the chance to do that. All right, so what I want you to do right now, just copy down here to just to the end of control. Don't copy the hypothesis part yet. Yeah. All right, so the first thing, obviously, when you're doing an experiment, the only reason you do an experiment is to investigate a problem. All right, so on the sheet that you get from me will be this problem, and it'll say something like, I don't know, hypothetically, what is the effect of thickness of plexiglass on its strength? Okay, that could be something that you might study. All right, if we want to do an experiment to test that, well, okay, what we would do is say, all right, well, our manipulated variable then needs to be what? The thickness of the plexiglass. Okay, my problem was, what is the effect of thickness on tensile strength? And what I need to manipulate, what I need to change, investigate, is the thickness of the glass. I need to have several different sheets of plexiglass that are different thicknesses. Okay. What's my responding variable? What am I hoping is going to happen the thicker I make the plexiglass? Right, the stronger it will be. All right, so my responding variable will be the strength of the glass. All right, everyone follow me there? Really? We'll get to that. Okay, just hang on for now. All right, so our manipulated variable is the variable that's changed by the experimenter. Our responding variable is the one that we watch. We're hoping to see it change as a result of what we did. So in the example we're looking at right now, I change the thickness of the glass, and I hope that as a result the glass gets stronger. So what I'm watching for is changes in the strength or the ability of the glass to keep from breaking. All right, so let's say that the experiment we're doing here is essentially we're taking like a .45 caliber pistol and we are firing bullets at this glass from 30 feet away. Okay, that's our experiment. All right, now I need to keep some things the same when I test the different thicknesses of glass. Agreed? If there's some things, if I don't keep them the same, my experiment isn't very valid. All right, what would some of those things be? Right, I need to be the same distance away from the sheets of glass in every trial. All right, it wouldn't be very fair if for the thickest sheet of glass I was 30 feet away, and for the thinnest sheet of glass I walked right up. Okay, that's not exactly a fair experiment. That glass has no chance from point blank range. Yes, I would have to make sure that I target the same part of the glass. Probably I would put an X in the center and have all the sheets of glass be exactly the same size and target the center portion of them in order to be fair. Because yeah, you might have a chance of breaking the edges off because there's less surrounding glass at the edges. Absolutely. I could use the same gun in every trial, right? It wouldn't be fair to walk up with a pellet gun on one and then have like a howitzer, okay, for the last one. That's not a fair test either. That's not controlled, okay? A test or an experiment that is fair and valid is referred to as a controlled experiment because all the variables that could affect the outcome are kept the same or controlled. Hence the name controlled variables. All right. What other things should I keep the same? Would temperature affect the strength of glass? Yeah, glass gets brittle when it gets cold. I wouldn't want to be testing these at different temperatures. All right. So all these kind of things have to be taken into account when you design an experiment. All right. Lots of times controlled variables are environmental things. Temperature, air pressure, and things like that. Sometimes it's amounts. All right, if you're testing something, you want to make sure that let's say you're testing 4 different substances and their effect on something. You want to make sure you test the same amount. All right, you don't want to test twice as much of one as the other. That's not really a fair test either. You want to make sure the amounts are the same. All of those things are controlled variables. All right, when medical companies test new drugs and things like that, that's one of the big things that they do, is make sure everybody gets the same amount. Okay? Now, when you run an experiment, one of the things that we sometimes overlook is that you have to have some way of knowing what was the effect of doing nothing. Okay? You have to have a comparison, what they call a control group. So when a medical company tests the effect of a new drug, they give everybody a pill. Just some people get what kind of pill? A fake one. A placebo is what it's called. It's a sugar pill. All right? But everybody gets to take a pill. And when a medical company does that, if the people who got the placebo, the sugar pill, get better too, what's the effect of their drug? It doesn't work. Okay? Or it works, but not significantly better than the effect of mind over matter. Sometimes just taking a pill makes you feel better. Okay? Really, there's lots of effects. There's lots of studies out there that show if you tell somebody this will make you feel better, they'll feel better. Okay? The mind is an amazing thing. All right? That actually happened lots of times back in like World War I and World War II. They'd run out of morphine, so they'd give the people a placebo and say, it's morphine, take it, you'll feel better. Oh, I feel so much better. Okay? Well, it works. Sometimes it's just mind over matter, right? Okay, so you always have to have a group that, let's say, is being experimented on, and a group that isn't being experimented on, so that you can see the effects of what you have done. All right. In our test here, our experiment with the types of glass, I should probably have as a comparison a sheet of plain glass. Obviously, we know what will happen if you shoot a piece of plain glass. It's going to break, but you still need to have that reference. All right. This is the effect of having no plexiglass. Just shatters. All right. Now I compare thicker and thicker and thicker plexiglass, and I find the effect. All right. So once you've identified your variables and you kind of set your experiment up, everyone knows what things they have to have in place and how they have to control them, then you'll put in your hypothesis. Okay? The hypothesis is the most important part of the experiment and is more than likely the part that people do wrong. Okay? For some reason along the way, people have probably told you that your hypothesis is just a guess. It is not. Okay? A hypothesis is not a guess. A hypothesis is a deductive, predictive statement. Right? Okay, so as I said before, a hypothesis is a deductive, predictive statement. So it has to deduce something. It has to say, based on these things, this is the solution. Okay, and then it needs to say, if that's true, these are going to be my results. All right. So that's why a hypothesis always contains the words if, and then. All right. It says the if part always talks about the problem. It is what you think the solution to the problem is, or the cause of the problem, okay, or something along those lines. The and part describes the experiment very, very briefly. The then part gives you the predicted findings. Right? Sometimes those are general, and sometimes they're very specific. Right? For our example here with the plexiglass, we could say this: if plexiglass gets stronger with increasing thickness, and a pistol is fired at sheets of plexiglass of varying lengths, then those sheets of then the thickest sheets will be less penetrated or will break less or something along those lines. There's here's what the relationship is, here's what I'm going to do, these are the results if I'm right. Okay? A hypothesis always goes in that order. Here's what I think the relationship is, here's what I'm going to do to test it, these are the predicted results. If I'm right, this is what I'll see. That way, when you're done the experiment, your conclusion is easy. I predicted these results. They didn't happen. I reject my hypothesis. Okay? Here's the results I predicted. These are the results I got. They're exactly the same. My hypothesis is acceptable because the data correlates with what I predicted. Right? Everybody follow me on that? So your hypothesis has to do those things. It has to say what you think, talk about what you're going to do, and tell people what you will see if you're right. All right? So if, and then. Okay? So I mean they can be any number of things. It could be like if I was testing the effect of caffeine on on learning ability, right? And I say if caffeine stimulates the brain, and half of a class is given caffeine, the other half is not, then the half of the class receiving the caffeine will do better on the quiz. All right. I mean, that would be a hypothesis. It's not a very good hypothesis, but it's a hypothesis. All right. It says what I think the effect of caffeine on learning is. It says what I'm going to do and predicts the results. Now, obviously, my experiment would have to be more detailed than give half of you caffeine and half of you not. All right. There'd have to be more to it than that. All right. Okay. The next thing that you'll have is materials. On your lab reports, you can always just write C sheet, because I'm going to give you a sheet that has the materials on it, and that is going to be the cover sheet for your lab report when you hand it in, because it already has your name on it, it's got the marking rubrics on it, all of that. So you don't need to rewrite it. You can just write materials, C sheet. That's as far as I want to go. I don't want people just keep copying. I want them to listen. Okay, and you can do the same thing for the procedure, unless the experiment asks you to design the procedure yourself, in which case you're writing out step 1, do this, step 2, do this. Okay, most of the time, with the exception of next week, okay, you'll just be C sheet. But next week, you're designing the experiment yourself, so you'll be writing out the steps. Once you perform the experiment, now what do you have? Before you have an outcome, you've got, what do you collect during an experiment? Data, right? You collect data, observations, those kinds of things. From lab to lab, those observations can be very different. Sometimes they're qualitative. The solution turned pink. Okay? Sometimes they're quantitative. The cart went 10.58 meters, right? Along those lines. But they're all recorded. Okay? You have all of this data. That's, again, very important stuff that needs to be included in your lab report. Where does it go? It goes under procedure. You write observations, and that's where you put in all of your data, whether it's a chart of some kind, whether it's anecdotal, which means you write in, you know, at 9 o'clock a.m. we did the following things, and the solution turned pink, and then it turned clear, and then it turned pink, and you're just writing it out as it happened. Okay, that's fine. But whatever your observations are, that's where they go. Okay, they go underneath procedure in a section called observations, okay, or data, as the case may be. You record them all. Okay. Once you have data, what do you do with it? You what? Okay, yeah, so we've recorded our data. We have it all here. What do I do with my recorded data? Starts with A. Starts with A. Analyze it. Right. Okay. So after your observations are your analysis. Sometimes that's 4 or 5 questions for you to answer. Sometimes it's, what does this mean? Okay? Sometimes the analysis is very simply, what does your experiment tell you? What does your data tell you? All right, so your observations are all your data acquired in the experiment, charts, graphs, measurements, okay, calculations. Sometimes your calculations will also be in your analysis. Okay, and then for your analysis, answer questions. And on a lab report, you do not need to rewrite the questions. The questions are on the front sheet that you're handing in, so there is no need for you to write out the questions, the analysis questions. Don't recopy them, just answer them. Should these be complete sentences? Always, all right? The answer to an analysis question is never yes or no. Okay? It is yes because, or yes in this way, the following thing, you know, whatever. There's always an explanation, right? But it's never yes or no. If you write yes or no and leave it at that, I guarantee you will not be getting 5 out of 5 on your analysis questions. All right? Okay. And then after your analysis, after you've looked at your data, and your data says, oh, this cart traveled the same distance every second, my conclusion would be then that this cart must have been traveling at a constant speed. All right, that could be my conclusion. Or if I'm back to the plexiglass experiment, right? At the end of my experiment, I find that the thicker sheets had less bullet penetration. All right, the bullet didn't go as far in and they didn't shatter. Well, when I analyze that, my analysis would be that thicker glass seems to have resulted in less penetration of the bullet. Now I can write a conclusion about that. Right? If that's what my data says, that's what I write in my conclusion. Okay? And the first thing you do in your conclusion is restate your hypothesis. So if you're doing this on the computer, which you are, because I don't accept these handwritten, right? Copy, paste. Okay? Copy your hypothesis, paste it back down in the conclusion, and then tell me why you accept or reject it. Right? Now, when you tell me why you accept or reject it, you have to tell me why. Yes, I was right is not a conclusion. I see it all the time, but it is not a conclusion. No one's ever 100% right in science. It doesn't work that way. Right? There are some parts that are right and some parts that weren't right. So you always have to say, this is the hypothesis. And based on these specific observations, observations say that thickness of plexiglass did increase its ability to resist bullets. All right? That is clearly shown given the data here. All right? That's what you need to say in your conclusion. This data says this, and that means my hypothesis is acceptable, or my hypothesis is rejected. All right? Everybody follow me there? Okay? So a hypothesis, that's going to be, 2-3 lines. Conclusion, it's going to be 5 or 6 lines, because you got your hypothesis, and then you got your explanation for why you accept or reject it. And then after that, at the very end, is what we call the evaluation. That is where you look at the experiment and tell me if there were sources of error that were not accounted for in the lab, that were beyond your control. Okay? They were, let's say, inherent to the experiment's design. I forgot to measure the temperature is not a source of error. I see that one all the time too. Sources of error. Didn't do this right. Oh, really? If you didn't do it right, you should do it again. Okay? Human error is not a source of error. Human error is a screw up. Fix it. Right? Because if I see on your lab report under sources of error, we didn't do this right, you get 0. And then I'm going to go back and find other places to dock marks because you obviously didn't do the experiment right. Okay? Don't tell me you screwed up. Fix it. Okay? Because what you tell me when you write in your evaluation, I messed up, is that you didn't care enough to go back and change what you did. You just went, crap. Screwed up. I'll just write that I screwed up. Too lazy to do it again. Okay? Your data is no good at that point, guys. All right? You have to go back and get valid data. All right, and like I say, I see it all the time. Like honestly, I see written on there in an experiment where taking the temperature was one of the key points of the experiment. Forgot to record temperature. Yeah, I don't know what to say about that, except that your experiment is really bad. Okay, and as a result, so is all of your analysis and whatever. People say I forgot to record the temperature, yet they have all this analysis about what the temperature did. Weird. Strange, right? So make sure that what you write in your evaluation is stuff that was beyond your control. All right. One of the things for our plexiglass experiment could be that we would probably have had no control over the manufacturing process involved in making the plexiglass. Could there have been flaws in the manufacturing process that we are not aware of? Yes. Okay. Beyond our control? Yes. You still put it in there. It's a possible source of error, right? You could have had one thick sheet that broke simply because it wasn't manufactured properly. Okay. If we're looking at the effects of the caffeine on the exam performance, okay, sources of error beyond my control in that one could be your tolerance for caffeine. If you're a person who drinks a pot of coffee a day and I give you a little tiny pill that only contains 10 milligrams of caffeine, it isn't going to have any effect on you. Right? Your body would have a tolerance built up for it. Right? If you're the kind of person who can drink a Red Bull and fall asleep, that little pill isn't doing anything. All right? It's not, it's beyond my control because I don't know your background and I don't know how many Red Bulls a day you drink. I hope it isn't very many. Okay? You guys follow me there? Those are things you put in. These may have been present, but they were beyond my ability to control in the experiment. They would have affected the experiment in this way. Okay? All right. Questions on how your lab report works? Okay. This is the order your lab report will always go in. It's always the order that it'll be on the sheet I give you. Okay, problem, restate it in your own words. Design, manipulated, responding, controlled, and why. Okay, hypothesis, materials, procedure, analysis. Okay, procedure would include your observations, analysis, conclusion, evaluation. Always goes in that order. Okay, is a lab report meant to be a ton of work for you outside of school? No. All right. Generally, guys, you should be able to get a lab report well done, half hour, okay, 40 minutes tops, and that's typed out. Right? You're going to get time, usually at the end of the lab, we have some time where we come back to the classroom because we're finished, and you get a little bit of time to work on it, okay, those kinds of things. So, okay, yes, it's a little bit of work outside of class time, but it shouldn't be meant as like a 4-hour thing. Okay. I will not accept them handwritten, no, they must be typed. All right, there are computers available in the school if you do not have one at home. All right, but they do need to be typed because proper lab reports. I mean, no one submits a scientific paper in their own handwriting. Okay. All right, so hang on to those guys. That's the important stuff there. Okay, there's always evaluation rubrics as well. Okay, just show you kind of what one looks like here. Okay, this is the one that you guys will be doing next week. Okay, so to give you an idea, all right, starts out the title of the lab, gives you a problem, okay, and then in the design it says identify your manipulated, responding, controlled variables and explain why you think this one's manipulated, this one's responding, right? Then you have your hypothesis, if, and then here's all your materials. You would write on your lab report, C sheet. Okay. Then in your procedure, for this lab, you actually write the procedure. So you'd say step 1, collect necessary materials. Step 2, put samples in depression plate. Step 3, record physical properties, whatever. Okay, like you would write in your step by step. If it's an experiment where I give you the steps, you just write C sheet. Right? Underneath that's going to be your observations. Probably for this one, it'll be a big table. Right? And then there's your analysis. Okay, your analysis is, tell me, was sample A ammonium nitrate, was sample B ethanol, okay, whatever. You copy this chart out and you fill it in. Okay, what was the purpose of testing distilled water? So you answer the questions, the rest of the analysis questions. Okay, and then you have your conclusion and your evaluation. Then you have your marking rubrics. Okay, you guys have all seen these before? Okay, there was a while where JP2 and us were using exactly the same rubrics. I don't know if that's still true or not. Okay, so you have your design. That's where I look at did you identify the material or did you identify the variables correctly? Okay, your hypothesis, 5 marks for getting the hypothesis. Okay, equipment and procedure. Sometimes this rubric will be on, sometimes it won't. For this lab, it is because you're designing the procedure. So I mark the quality of your procedure on this one. Okay. 3 marks for your data. Okay, is it good? Is it organized? Is it accurate? Okay, your analysis, 5 marks for that. Sorry, this is an old version. 4 marks for conclusion now, not 3. 3 marks for evaluation, and then the overall quality of your report is 5 marks. Okay, is it go in the right order? Is it easy to follow? Is it neat? Okay, you know, those kind of things are part of the overall. Okay. Everybody good with that? All right.